In this video, we are going to learn about the colloidal system in detail, major points about this particular topic. We will be just reading the topic because this is not that much complex to learn. We will be just reading the topic. So, which are the major points? So, first point is the diffusion. Like soluble particles, colloidal particles diffuse at a slower rate from a region of higher to a region of lower concentration. Next, there will be sedimentation. The colloidal particles tend to settle down very slowly. Then solvation is happening. Non-dialyzable property will be there and gelation will be happening. So when we just learn about the gelation property in detail, we can see that there are exothermic gel and there is a liberation of heat and fall in internal energy or gelation of gel. Gelation occurs both in lyophilic and lyophobic sols. Though it contains significant portion of liquid, it possesses a greater or lesser degree of rigidity. Gelation is caused by the development of a very definite structure within the dispersed system that is brought about by the inherent adherence of some of the colloidal particles to one another to form fibrillar network to enmesh a more dilute colloidal solution by capillary action and partially by hydration of the fibrils. Examples of gels are fruit jellies, cooked starch, and heat coagulated egg white. Later on, a little volume of liquid diffuse out of the gel for shrinking and condensation of the gel mass due to the gradual shortening of the fibrillar threads. This is called stringencies. Synergesis. Synergesis. Syneresis. The example of syneresis is that of the blood clotting. Serum diffuses out of the clot due to shrinking and condensation called clot retraction or syneresis. Syneresis phenomenon will be happening. And weeping of the gel or aging of the gels, which can be regarded as the reverse of the absorption which previously took place in the gel formation. Next, inhibition or quelling. Many colloids such as starch or gelatin, which are insoluble in cold water, absorb water and exhibit a phenomenon called imbibition. It's about imbibing, swelling reaction. Then there will be viscosity. Regarding viscosity, lyophobic colloids do not show much difference from pure solvents, but lyophobic colloids do not show much difference from pure solvents, but lyophobic colloids won't be showing difference, but lyophilic colloids have higher viscosity than the pure solvents because of big size of colloidal particles, solvation around the particles, and much of the liquid engaged for the process, zeta potential around the dispersed particles, elongated and coiled shape of the dispersed particles. All these are the reasons for this higher viscosity in case of lyophilic colloids. Next is the protective action. Certain colloids may exercise a protective action to the other colloidal solution. The addition of lyophilic colloid, which is highly stable, to a solution of lyophobic colloid solution prevents or delays the coagulation and imparts stability to the lyophobic colloids. This is known as the protection, and the colloid is called as a protective colloid. What is a protective colloid? A colloid, something which is added into, this is a type of lyophilic colloid that is going to be added into the lyophobic type. Lyophobic type is an unstable one, so to stabilize that and to prevent coagulation, we will be adding a protective colloid into that. Colloidal gold is much more stable in the presence of little gelatin. It is due to the adsorption of protective colloid by the sol particles. Next, peptidization will be happening. Specific substances in small amounts called peptizer, which change the insoluble solid of low molecular weight, which may be a coagulum or a precipitate or any other form, into the lyophobic sol called the extrinsic colloid. Many ions in low concentration act as peptizer, such as AgCl precipitate is baptized by Ag plus or Cl minus ions. Next is the thixotrophy. Thixotropy like THIXOTROPY. Mechanical agitation may change a gel to a solid. This is due to lowering of viscosity. This mechanism is known as thixotropy. What is that? Thixotropy. Thixotropy. When agitation ceases, it solidifies again. Practically all colloidal solutions show thixotrophy, but the favorable result is seen in case of rod-shaped missiles. These particles link up with each other to give mesh-like structures, but this weak linking cannot stand the stress by mechanical agitation. Exposure of cell to magnetic field shows thixotropy. Thixotropy will be happening. When we just expose this colloid to magnetic field, the thixotropy will be happening due to the lowering of viscosity. That is a change from a gel to a solid. Next is the adsorption. Adsorption is another property of colloids. Colloidal particles called adsorbent adsorb substances called absorbate on their surface, such as colloidal plasma protein adsorb mineral ions, sugar, lipids, etc., and carry to other planes. Next is the optical properties. Now we are going to learn about the optical properties of colloids in detail. First is the Brownian movement. We have already learned about Brownian movement in our earlier classes, but if you are first time learning this, please listen. The English botanist Robert Brown in 1827 observed that pollen grains dispersed in water exhibited rapid movement in all directions, rendering impossible to count their number in the field under microscope. Not normal microscope, it must be an ultra microscope. Later, this random zigzag motion was observed in all colloidal particles, including colloidal particles suspended in gaseous medium. Smaller particles exhibited more rapid and brisk motion than the later ones. Such movements are called Brownian movements. Brownian movement cannot be initiated by agitation, heat, and light. The cause of Brownian movement is due to the moving molecules of the dispersion medium constantly colliding with the colloidal particles caused by the thermal motions of the liquid molecules and impart momentum to the colloidal particles. This movement is strong evidence in support of the kinetic molecular theory of liquid and gases. Brownian movement is very important. Ultramicroscope studies have shown that some of the granules present in the living cells move about within the cells and in certain cases display Brownian movement. Muscle cells, leukocytes, etc. They show the Brownian movement. Zigzag type of movement, Brownian movement of colloidal particles. Next is the Tyndall effect. Tyndall effect or phenomenon of Faraday-Tyndall phenomenon. What is that Tyndall effect? We will be learning about the Tyndall effect in detail. The presence of colloidal particles are not detectable under a microscope but can be made visible by optical means. When a beam of light is passed through an optically homogeneous solution, the course of the beam is not visible. When a beam of sunlight is falling on a dark room, the dust particles in the path of the light is visible due to scattering of light from their surface. Thus, if an intense beam of light is passed at right angles through a colloidal solution, the particles in scatter light and beam is rendered visible and the individual particles can be encountered. This phenomenon is known as the Tyndall effect, and the apparatus using the principle of dark ground in illumination is called ultramicroscope, constructed and used by Zsigmondy. In the limit of the Tyndall effect may be as a test for presence of ultramicroscopic particles having size between 5 mu to 50 mu. Tyndall showed that if the particles were sufficiently minute, the light dispersed by them at right angles to the beam was polarized and then again tested by looking into the beam through the Nicol prism. Not all colloidal solutions show such particles in ultramicroscope. Lyophobic salts only show this effect, but not the lyophilic salts. The reasons are lyophilic colloids are the ones which show this. Lyophobic salts. Lyophobic salts will be showing. The reasons why lyophilic salts won't be showing this tyndall effect is that the absence of optical heterogeneity, which may be due to coincidence of refractive indexes of hydrated colloids and water, absence of any clear dividing surface between the two phases. It can be stated that it is not the colloidal particles itself which is rendered visible, but only the light scattered from it. Small size of the particles and addition of any substance causing aggregation and therefore increase the size of colloidal particles render them into view. Next, let's learn about the electrical properties of this colloids. First is the charge, electrical charge. The colloidal particles, both lyophilic and lyophobic. Wait a minute. And we just learned about the Tyndall phenomenon. So, which type of colloids show this Tyndall effect? Yes, lyophobic salts only show this effect. Why? Because lyophilic salts can't show them because of two reasons. First one is the absence of optical heterogeneity, and next one is the absence of any clear dividing phase between the two phases. That are the reasons which are responsible for lyophilic salts to not to show the Tyndall effect. Next, electrical charge is the electrical property. The colloidal particles, both lyophilic and lyophobic, carry an electric charge, either positive or negative. The charge upon the particles is important in stabilizing colloid solutions because all the particles in a given sol are charged alike and repel each other and remain in suspension. The neutralization of this charges has importance in the precipitation or flocculation of colloids. The neutralization of these charges has importance in the precipitation of flocculation of colloids. The electrical charge of colloid particles arise from the ionization of groups in the particle surface like protein through selective adsorption of ions from the medium upon surfaces. Next, both the mechanisms may act together. Double layer of colloidal particles or Helmholtz gave double layer theory. This theory explains the charge relations at the surface of the colloidal particles. According to this theory, the surface of the colloidal particles are negatively charged, and this is surrounded by oppositely charged ions, positive ions. Colloidal particle surface is negatively charged, then they are surrounded by positive ions. There are two layers of positively charged ions. An immobile layer of ions from the liquid from the liquid absorbed on the surface, more diffuse layers of mobile ions in liquid around the particles. The mobility of mobile layer increases according to the distance from negatively charged surface. But if the colloidal particle is positively charged, the relations of the double layer are opposite and the surrounding liquid remains negative ions. So when colloidal particle is viewed with the help of an ultra microscope, we can see colloidal particles. Halo of scattered light around a colloidal particle will be seen. When the Helmholtz double layer and potential can be seen, like the epsilon potential is applied, there will be zeta potential. Then when we just stem potential is also there. So stem potential gives negatively charged surface of the colloidal particles. Then B type gives immobile layer of ions from the liquid adsorbed on the surface. More diffuse layers of mobile ions in liquid around the particles. Epsilon potential or electrochemical potential. The potential drop across all the ionic layers from the negatively charged surface of the particle up to the mobile positive ions into the solution is the epsilon potential. Epsilon potential. There is a structure potential called epsilon potential. Next there will be zeta potential. Zeta potential is another term, important term. The potential between the immobile positively charged layers of ions and the mobile phase of positive ions is called a zeta potential or electrokinetic potential. There will be epsilon potential is there. Epsilon, next zeta potential, next this Stern potential. The potential between the immobile positive layers of ions and the negatively charged particle surface is called Stern potential, and it is equal to epsilon electrochemical potential minus zeta potential electrokinetic potential. What is zeta potential? It is electrokinetic potential. Electrokinetic. But epsilon electrochemical potential. Electrochemical potential. Electrochemical potential. That is the epsilon potential, electrochemical potential, and zeta potential is electrokinetic potential. Next, there is a Stern potential. Stern potential. Stern potential. How to calculate this Stern potential? Equal to epsilon potential minus zeta potential. That is electrochemical potential minus electrokinetic potential. Epsilon potential is equal to Stern potential plus zeta potential. Epsilon potential is equal to Stern potential plus zeta potential. Among these potentials, the zeta potential is something which is represented by the electrokinetic potential. Zeta potential is important due to the properties of colloids. By addition of electrolytes, there is a drop of zeta potential to a certain level called critical potential. When we add electrolytes into the zeta potential will be dropping. So that is called as a critical potential, and the double layers of the colloidal particles do not persist and there is aggregation of the particles and precipitation occurs. That is happening when we add electrolytes. Zeta potential is very important in this. There will be a drop of zeta potential when we add electrolytes, and after that process the critical potential will be developing and precipitation occurs. So can we just look at the streaming potential is also there. Streaming potential, like glass capillary walls are there and in between water will be there. So streaming potential can be seen. Streaming potential or flow potential is the next topic. A glass capillary contains water. The walls of the capillary become negative and the water is positive. If the water is forced to flow through the glass capillary, a potential is developed between the two ends of the capillary, and this potential is known as streaming potential or flow potential. The explanation is that the capillary walls absorb ions from the water, leaving H plus ions which charge the water molecules positively. These positively charged water molecules are mobile and accumulate in excess at one end while flowing through the capillary, leaving excess negatively charged molecules at the other. This develops a potential difference between the two ends of the glass capillary. Difficulty is encountered during filtration through this and fine pores due to the establishment of strong streaming potential in the membrane pores. This is observed particularly in case of solutions having low electrolyte concentration because electrolytes reduce the potential at the pore surface. Wider reduction of zeta potential on the surface of colloids by electrolytes. So this is about the streaming potential. Streaming potential or flow potential. These are the two terms. Streaming potential or flow potential. Biological importance. Probably the flow of fluids to the cell membranes or tissues produces streaming potential, which is a contributory factor for the potential difference across the membranes. The electrically charged pore surface of filters is important for accessing the impermeability of the filters to bacteria and other microorganisms, which is usually negatively charged. The negatively charged pore surfaces of filters is important for accessing the impermeability of the filters to bacteria and other microorganisms, which are usually negatively charged. Next term is the electroosmosis. The movement of the dispersion medium under the influence of the electric field is called electroosmosis. This can be observed by fixing a porous diaphragm made out of material dispersed phase in a U-tube containing two portions of the liquid dispersion medium as shown in the diagram. When an electric field is set across the ends of the tube by placing the electrodes on either side of the diaphragm, the solid particles and the fixed layer of charges cannot move through the diaphragm, but the liquid like dispersion medium and the diffuse charge layer of this dispersion medium can move through the diaphragm towards oppositely charged electrodes compared to the charges in the diffuse layer. Next, learn about the electrophoresis or cataphoresis. Colloid particles are electrically charged. So the colloidal unit, both lyophobic and lyophilic, move in an electric field either towards the cathode or towards the anode, depending on whether they are positively or negatively charged. This phenomenon of movement of colloids in an electric field is known as cataphoresis or electrophoresis. The direction of migration of particles depends upon the sign of the charges. The speed of migration is about 1 micron per second per unit potential gradient. The rate of migration varies directly with the net amount of the charge, inversely with the size of the particle and the viscosity of the dispersion medium. During the process, there is a potential difference existing between the surface of the colloid and its neighboring solution, which is called the zeta potential, and its value is 50 millivolts. Some scientists developed the electrophoresis method of protein analysis and applied it to a study of plasma proteins, and the apparatus used for this study is known as Tisselger's apparatus. Next is the ampholytic character of amphoteric substances like ampholites. Lyophilic colloids such as proteins, albumins, globulins, gelatin, etc. behave as an acid in alkaline medium and as a base in acid media. Hence, this is called ampholytic or amphoteric behavior. Amphoteric character. Let me just represent to a U-tube with plus sign and minus sign on the two ends. There will be porous diaphragm. In between these two regions, there will be flow occurring between the positive to negative side. Then negatively charged colloidal particles of the colloid migrate to the positive electrode and after losing charge are deposited on it. That is happening in the electrophoresis. It is electrophoresis. We will be representing a U-tube. A U-tube will be there, and a positive sign will be there into one end. Negative charge will be there on the other end. Ions will be moving into the positive charge for just illustrative purpose. And that is about electrophoresis. Next, we have learned the amphoteric property, right? So they are called ampholites because they carry both positive and negative charge. This is due to ionization of the molecule. The proteins contain amino acid and it has NH2 plus group and COO minus group. This amphoteric nature is due to NH2 plus and COO minus. Okay. And they are positively charged in acid media and negatively charged in alkaline media. Next property is the isoelectric pH or isoelectric point. Isoelectric pH or isoelectric point. Whether a protein has a positive or negative charge depends chiefly on the hydrogen ion concentration or pH, because pH has the influence on the ionization of the amino acids of protein. Due to the ampholytic character of colloid during cataphoresis at a particular pH, the colloidal particles does not move to the anode or cathode. At this point, no cataphoresis takes place. This pH of electrical neutrality of the molecule is known as the isoelectric point or isoelectric pH. At this point, following events are observed: complete ionization of both groups according to the switcher ion theory. Complete ionization will be happening. Complete ionization of both groups according to the switcher ion theory. Complete isolation will be happening. Ion carries complete ionization of both groups according to the switcher ion theory. Ion carries equal charge of positive and negative charges, hence has a net charge of 0, carries no charge, often shows flocculation, exhibits maximum precipitability, minimum viscosity. This is something which we have experienced during our laboratory works, because when a protein approaches its isoelectric point, there occurs the process of flocculation or precipitation that exhibits maximum precipitability and minimum viscosity will be there. Minimum osmotic pressure will be there, minimum imbibition will be happening. Imbibition, no cataphoresis. There will be zero charge will be there, and minimum viscosity will be there, often shows flocculation, exhibits maximum precipitability, and then maximum precipitation will be happening. Minimum viscosity, minimum osmotic pressure, minimum imbibition, no cataphoresis. That is happening in case of protein. Like protein will be centered and it has CO group on one side and NH2 plus groups on the other side. That is happening at isoelectric point. Net charge is 0. Some other things are also 0, like there will be not 0, minimum viscosity will be happening and minimum osmotic pressure will be there, minimum imbibition happening, no cataphoresis, and flocculation will be happening. Switcher ion theory is taking into account like complete ionization of both groups. So major points about isoelectric point is that this is a pH where the colloidal particles does not move to anode or cathode. At this point, the cataphoresis will not be taking place. So electrical neutrality is maintained in this isoelectric point. There are major points like precipitation will be happening maximum in this point, flocculation will be happening, there won't be any cataphoresis, there is no, not that much, minimum viscosity is there, complete ionization of both groups according to the switcher ion theory. Ion carries equal number of positive and negative charges, hence the net charge will be 0. Viscosity will be the minimum and minimum osmotic pressure will be there, minimum imbibition will be happening and no cataphoresis. So that's about the isoelectric point and related pH. Precipitation of colloids at their isoelectric point is an important method of purification of colloidal substance and alternatively of removal of colloidal impurities from the mixture. Switcher ion or dual or bipolar ions. Amino acid present in the protein molecule such as an amino acetic acid, glycine, shows interesting ampholytic property. It has both acid and base portions and can ionize in two ways. If ionization takes place in two ways from the same molecule, then an ion having both positive and negative are left. Such ions are called switcher ion or dual or bipolar ions. An amino acid may exist in aqueous solution in isoelectric point as switcher ions. The switcher ion theory can be applicable to polypeptides and proteins. Next is the Hofmeister series and colloids. When gelatin is immersed in aqueous solutions, its swelling is harnessed by NaCl, NaBr, KCl, NH4Cl and NaNO3 and retarded by sodium sulfate, tartarate, citrate and acetate. The action of ions on colloid protein is a complex phenomenon since both positive and negative ions exert an effect which is independent on the nature of the ions. It can be arranged for certain ions in the order of their effects in affecting the phenomenon and other properties of colloids. Arrangement in order of series is called the Hofmeister series, which is as follows, like when we arrange CN will be